Assalamu alaikum. Dear students, I'm here, Hafiz Muhammad Ammar, co-teacher for course Microbiology, code BOTN 4128. Today we are here for lecture number 20 entitled Growth of Microbial Cultures. Let's have a look at contents. Today we will discuss microbial growth redefined, cell division, either by binary fission or sometimes by budding, and sporulation. Microbial growth redefined. In everyday language, growth refers to an increase in size. We are familiar to seeing children, other animals, and plants grow. Unicellular organisms also grow, but as soon as a cell called the mother cell or parent cell has approximately doubled in size and duplicated its contents, it divides into two daughter cells. Then the daughter cells grow and subsequently they also divide. Because individual cells grow larger only to divide into two new individuals, microbial growth is defined not in terms of cell size but as the increase in number of cells which occurs by cell division. Growth may be defined as an increase in cellular constituents. It leads to a rise in cell number when microorganisms reproduce by processes like budding or binary fission. Growth also results when cells simply become longer or larger. If the microorganism is cynocytic, that is a multinucleate organism in which nuclear divisions are not accompanied by cell divisions, the growth results in an increase in cell size but not cell numbers. It is usually not convenient to investigate the growth and reproduction of an individual microorganisms because of their small size. Therefore, when studying growth, microbiologists normally follow changes in the total population number. Cell division. Cell division in microbes, unlike cell division in eukaryotes, usually occurs by a process of binary fission or sometimes the process of budding or sporulation and other methods including genetic recombination where genetic informations are exchanges, conjugation method, transduction method, or transformation methods. Let's have a look at binary fission method. Here are 4 steps involved. If we look at the step 1, which is known as replication of DNA, the bacterium uncoils and replicates its chromosomes, essentially doubling its contents. The step 2, after the copying the chromosomes, the bacterium starts to grow larger in preparation for binary fission, which is followed by an increase in cytoplasm volume as well as increase in the number of organelles. Other prominent trait of this stage is that the two strands migrate to opposite poles of the cell. In the step 3, segregation of DNA occurs. The cell elongates with a septum forming at the middle. The two chromosomes are also separated in this phase. In step 4, a new cell wall is formed at this phase and the cell splits at the center, dividing the parent cell into two new daughter cells. Each of the daughter cells contains a copy of nuclear materials as necessary organelles. Here is the example shown for binary fission, where you can see in upper part of the picture cell wall, cell membrane and elongated nuclei which is followed by septum invasion into the cell, and the nuclei is divided into two parts. And this septum, transverse septum, becomes complete, and two daughter cells are been separated in the next phase. Here you can see, if we study in stepwise, a young cell at early phase of the cycle, where you can see the organelles, nuclear material, and the cell structures. A parent cell prepares for division by enlarging its cell wall, cell membrane, and overall volume increases. Septum begins to grow inwards as the chromosomes move towards opposite ends of the cell. You can see in the third picture. Other cytoplasmic components are distributed into the two developing new cells, and septa is initiated at the center of the cell. Then the septum is synthesized completely through the cell center, and the cell membrane patches itself so that there are two separate cell chambers are formed. At the last point, the daughter cells are divided. Some species separate completely, while others still remain attached, forming chains, doublets, or cellular arrangements in other organisms. So this is the process of binary fission. I hope you understand this method by these pictures, illustrations. Another technique is budding. In this process, a small bud forms at one end of the mother cell, or on filaments, that's called prosthecae. As the growth proceeds, the size of the mother cells remains about constant, but the bud size enlarges. When the bud is about the same size as the mother cell, it separates from the mother cell. For example, in the Brewer's yeast, you can see in this yeast. Here is a pictorial representation of the budding. You can see in the picture, it shows the budding in yeast. Cell division in yeast and a few bacteria occurs through budding. In that process, a small new cell develops from the surface of an existing cell and subsequently separates from the parent cell. In this picture, you can see the reproduction by budding in yeast. In step 1, you can see small cells portion enlarges towards to make a bud. As bud develops in step 2, you can see, and when bud enlarges and have equal amount of cell material, organelles, nucleus, etc., the new bud then starts separating from the parent bud, or it may also be in the form of attached chain buds, and the process continues. Sporulation. Many environmental bacteria are able to produce stable dormant or resting forms as a branch of their life to enhance their survival under adverse conditions. Such dormant forms are called endospores, cysts, or heterocysts. Examples include gram-positive groups of bacteria, aerobic rod bacillus cells, microaerophilic rod bacteria. Cell division with the method of sporulation starts in response to nutritional deficiencies or nutritional deprivations, and these endospores have no metabolic activity through the process. When nutrient fulfills, it differentiates into vegetative cells. One spore formed in the bacteria at a time and begins with invagination of membrane around a small part of bacteria chromosome and generate an endospore or called cyst. Stage wise representation of sporulation method. Stage 1, you can see vegetative growth under unfavorable conditions. Stage 1 called axial filamentation and chromosome division. Activation of the master transcriptional regulator is followed by chromosome replication with each chromosome moving to the opposite poles of the cell. One of the chromosomes remains in the mother cell while the other is contained in the forespore. A forespore is a section of the cell containing chromosome. In stage 2, asymmetric septation occurs. Proximal regions of the cell are tethered to the opposite poles of the cell. Chromosomal DNA stretches, forming axial filaments. In stage 3, engulfment. As the cell divides, 30% of the origin proximal portion containing chromosome is covered. This is known as the forespore. A section of the cell containing chromosome is engulfed by a membrane as the peptidoglycan is synthesized. In stage 4 to 5, cortex and coat assembly, synthesis of the peptidoglycan cortex followed by the formation of proteinaceous spore coat. In this stage, the spore mature is enclosed in two different layers that include the outer coat consisting of different types of proteins and the cortex that consists of special type of peptidoglycan. We will show this later in the lecture. In next stage, the lysis of the mother cell occurs. The mother cell undergoes lysis. Some material from the mother cell may be used to build on the spore coat. In the next stage, spore releases as matures into the environment and are capable of surviving high temperature, certain chemicals that can destroy the vegetative cell, and radiation among the other extreme environments. Once environmental conditions improve, the spore can germinate and organisms returns to its original vegetative growth. In the 19th century, a physicist named John Tyndall discovered endospores, and these are highly resistant, non-reproductive, metabolically dominant structures produced by bacteria commonly found in soil and water. Spore formation occurs in bacteria in approximately 6 to 8 hours after being exposed to adverse environmental conditions. Mature endospores are released from normal growing cells that are called vegetative cells when spores are exposed to favorable conditions that supports their growth. They can germinate into a vegetative cell within 90 minutes. Endospore formation and cell cycle location. The location of spore varies in different species. Endospores may be present in the middle of the bacterium, for example in Bacillus cereus, at the end of the bacteria, also called terminal portion, for example in Clostridium tetani, the pathogen that causes the disease tetanus, and the in near end of the bacteria, also called subterminal end of the bacteria. The shape of spore also varies in different species. Endospores may be spherical, oval, or spore shape may also be diagnostic use. Here is the structure of the bacterial spore. An endospore is structurally and chemically more complex than the vegetative cell. It contains more layers than vegetative cells. The arrangement of spore layers is as exosporium. The spore is sometimes surrounded by a thin covering which overlies the spore coat. Spore coat is resistant to many toxic molecules. It acts like a sieve that excludes large toxic molecules like lysozymes. Spore coat may also contain enzymes that are involved in germination. Cortex consists of peptidoglycans and present beneath the spore coat. And you can see in the picture. Core wall lies beneath the cortex and surrounds the core of the endospores. Core contains the spore chromosomal DNA, which is encased in chromatin-like proteins known as small acid-soluble spore proteins. These are tightly bind and condense the DNA and protect the spore DNA from UV radiation, heat, and DNA damaging chemicals. Within the core, endospore consists of calcium dipicolinate, which is thought to stabilize the DNA. The resistance of bacterial spore may be mediated by dipicolinic acid, and calcium may aid in the resistance to heat and oxidizing agents. Difference between the cyst and endospores. Cyst and spores are resting structure of microorganisms. Unlike cysts, spores are reproductive structures that can develop into a new individual. So this is the key difference between cyst and spore. Further, encystment is the formation of process of cysts, while sporulation is the formation process of spores. Therefore, we can consider this also as a difference between cyst and spore. Further difference between cyst and spore is the cyst formation predominantly occurs under different types of adverse environmental conditions, but sporulation mainly occurs under nutrient deprivation. A cyst is a dormant state of microorganisms. It facilitates the survival of microorganisms under unfavorable environmental conditions. Encystment is the process by which the internal parasites, mostly in larval stages, residue resides within a cyst. Spore is another dormant structure produced by microorganisms mainly under nutrient deprivation. But unlike a cyst, spore is a reproductive structure which can develop into a new individual. However, both cyst and spore help microbes to withstand unfavorable conditions. Moreover, they contain resistant outer covers. Therefore, both structures cannot eliminate easily by many sterilization methods. So here is the reference material where from this lecture was prepared. You can also study this material. It is available in the web. Thank you very much.